Welcome to the Deep Dive, where we break down the most groundbreaking research.
And today we have a really big one, a story of incredible genomic precision.
Right, in the brain, the single biggest finding here is just, it's stunning. Researchers used prime editing, the super high precision gene editor,
The gold standard for precision.
and they used it to correct a disease-causing mutation inside the brain cells of living mice.
And this wasn't just some small effect. The correction actually rescued these mice from a severe fatal neurological disorder. It dramatically extended their lifespan.
So let's get into the target here, because the disease itself is a huge challenge.
It's a devastating one.
Yep.
It's called alternating hemiplegia of childhood, or AHC.
And that involves recurring paralysis, seizures. It's severe.
Extremely. And in most cases, we know the culprit. It's specific mutations in a gene called ATP1A3.
Which encodes a really critical ion pump in the brain, the Na plus K plus ATPase.
Exactly. It's one of the engines that makes the brain work.
And this isn't just a case of the pump not working.
Right.
It's a dominant negative mutation, right?
That's the key. That's the whole molecular hurdle. The faulty protein that's made doesn't just sit there. It actively interferes with any healthy protein.
So you can't just add a new healthy copy of the gene.
Nope, that won't work.
Yep.
You have to go in and correct the original toxic copy.
And doing that in neurons is tricky. They don't divide. So standard CRISPR with its double-strand DNA breaks is just too risky.
Way too risky. You could get all sorts of chromosomal problems, toxicity. It's a non-starter in the brain, really.
So prime editing avoids that. It just nicks one strand of the DNA.
Right. It minimizes the risk, but still lets you make the precise base pair swap you need for this AHC mutation.
Okay, so the tool is right, but the delivery, getting that huge prime editor complex into the brain, how did they solve that? I think figure 3 really covers this.
They did it with a really smart strategy. They used something called dual AAV9 vectors.
AAV9, that's the one that's neurotropic. It homes in on the nervous system.
Exactly. They split the editor into two separate viruses and then injected them together directly into the brain's fluid cavity in newborn mice.
And what were the results? Molecularly, did it work?
It worked incredibly well. They saw up to 48% DNA correction across the entire brain cortex,
Wow.
and this directly restored function. The untreated mice, the D801N models, they only had about 59% of normal ion pump activity after the treatment.
What did it jump to?
It jumped to 87% of wild-type activity.
87%. That's almost normal function from a single injection.
It really is.
Okay, so you fix the pump, the molecular problem is solved, but here's the real question, the one that really matters. Can one single genomic fix that early in life actually reverse these complex, devastating neurological and behavioral problems, for the long haul?
That is the core question. And the answer, which we see in figures 4 and 5, is
Yeah.
a pretty resounding yes.
Okay, so what did they see?
Let's start with survival. The untreated male mice had a median survival of just 13 weeks
13 weeks.
after the prime editing treatment. 56% of those males survived all the way to 52 weeks, a full year.
That is a massive, durable extension of life. But was it a good life? Did it improve their symptoms?
It was a full-on multi-phenotype rescue. They saw a huge reduction in those paralysis and seizure-like episodes and their motor coordination on a rotarod test.
Let me guess. It improved.
It was completely rescued, back to the same level as healthy wild-type mice. The treatment basically normalized their neurological health.
And figure 6 really drives home why they needed this high-precision approach. They tried a different therapy too, right?
They did, just as standard gene addition. They used AAV9 to deliver extra copies of the healthy ATP1A3 gene.
And the result?
Nothing. No improvement at all. Not in behavior, not in survival. It proved that for this kind of disease, you have to correct the original gene.
This really feels like a major milestone then. It shows that this kind of nick-based editing can safely and effectively fix a neurological disease in vivo.
Absolutely. It puts prime editing right in the clinical spotlight. This could be a template for a one-time permanent cure for so many other genetic brain disorders.
Especially things like channelopathies, where you just can't accept the risks of older CRISPR systems.
Exactly.
So to wrap up, let's hit our 3 key takeaways. First,
Prime editing worked. They got it into CNS neurons with AAV9 and successfully corrected the gene.
second, that correction had real-world consequences. It restored the pump's function and rescued behavior, coordination, and, critically, survival.
And third, the precision was absolutely necessary. Simple gene addition failed, proving that for dominant negative diseases like this, you need to edit.
Right. Now, this is a huge proof of concept, but obviously there's more work to do.
Oh, for sure. We still need to figure out the best treatment window and really confirm the long-term safety across different parts of the human brain.
But this paper tells us that it is, in principle, possible to rewrite the code of an ion pump in enough neurons to calm a chaotic brain. So looking forward, what do you see as the next critical hurdle to get this kind of precision to patients?
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.